What's the difference between CNVs and SNPs? Have you ever wondered how tiny changes in our DNA can impact our health? Today, we are going to break down the differences between copy number variants and single nucleotide polymorphisms, especially in relation to schizophrenia. Let's start with the basics. Single nucleotide polymorphisms, often called SNPs, are variations at a single nucleotide position in the DNA sequence. This means that one base, which can be adenine, thymine, cytosine, or guanine, is replaced by another. These are the most common type of genetic variation and can occur frequently in the population. On the other hand, copy number variants involve larger segments of DNA. These segments can be thousands of base pairs long. In CNVs, sections of the genome are either duplicated or deleted, leading to different numbers of copies of particular genes or DNA regions in individuals. Now let's talk about scale and impact. SNPs only affect one nucleotide. Depending on where they occur, they may or may not influence gene function. In contrast, CNVs affect larger segments of DNA, often including whole genes or multiple genes. This can significantly alter gene dosage, which is the number of gene copies, and can have a more dramatic impact on gene expression and function. When it comes to schizophrenia, CNVs have been identified as important risk factors. Certain recurrent CNVs are found in about 3.5 to 5% of schizophrenia cases. While these CNVs can increase the risk of developing the disorder, they are not the sole cause. Their expression can vary and they may show reduced penetrance. SNPs also contribute to genetic risk for schizophrenia, but they typically representing more subtle variations. While SNPs are widespread, CNVs often have a stronger effect because they can disrupt multiple genes or regulatory regions. Let's consider the functional consequences. CNVs are more likely to affect protein-coding genes and gene dosage, which can influence brain development and function. This is particularly relevant in schizophrenia, where changes in gene dosage from CNVs may contribute to neurodevelopmental abnormalities. SNPs, depending on their location, can affect gene regulation or protein structure, but usually on a smaller scale. In practical terms, identifying CNVs in individuals can help understand their genetic risk profile for schizophrenia and related neurodevelopmental disorders. CNVs are also linked to other conditions such as autism spectrum disorder and intellectual disability, indicating overlapping genetic influences. Genetic testing for CNVs can be part of research and clinical assessments to better characterize the biological basis of schizophrenia and potentially guide personalized approaches. So, to sum it up, single nucleotide polymorphisms are small-scale, single-letter changes in DNA that are common across populations. Copy number variants are larger structural changes involving gain or loss of DNA segments. Both contribute to genetic diversity and disease risk, but CNVs play a particularly notable role in schizophrenia due to their impact on gene dosage and brain-related genes.